So today we are going to talk about basic bacteriology, general bacteriology. And some of the stuff may look familiar to you because it's coming from biology. And I don't expect myself to spend much time on that. You need to know differences between human and bacterial cell. What are the classification methods that we have? And then structure, chemical constituents, and function of cell wall. This is going to be the most important thing for today's discussion. Now, just to recap, you can see from here prokaryote and eukaryote. So there are some subtle differences between these two. And the whole idea is that we really have to understand how bacteria multiply, how they grow, and how they function before we use any of the antibiotics that we normally do. And this is basically the upshot for the differences between eukaryote, that includes us as well, algae, fungi, plants, and animal, and in prokaryote, you only have bacteria. And if you look at the size of it, 5 micrometer, mostly bacteria are less than 5 micrometer. So if you were to use a sieve, like a filter in the lab, to get rid of those bacteria, you have to have at least a pore size bigger than 5 micrometer. The most important thing that I want you to pay attention is the nucleus. There's a classical membrane as we have, as compared to no nuclear membrane in bacteria. As far as chromosomes are concerned, there are strands of DNA, diploid genome, very commonly asked question and very important, as compared to single circular DNA, which is haploid. So this is the major difference that we have. Then you can see from here, mitochondria, Golgi bodies, and endoplasmic reticulum are pretty much absent over here as compared to other eukaryotes. If you look at ribosomes basically are the protein manufacturing plants, and you will see again, and these are the targets of antibiotics that we normally have. Cytoplasmic membrane, it contains sterol, it doesn't contain sterol. Cell wall basically is present in fungi, otherwise absent. And cell wall basically is the most complex structure that I'm going to spend some time today, and that contains pretty much proteins, lipids, and peptidoglycans. Bacteria divide very fast, they're very quick, and by the time we go after them, they may have multiplied thousands of fold. And they have a division pretty much asexual binary fusion, as compared to us and other plants, fungi, and protozoa have sexual. And sometimes in some plants and other protozoa, we may have asexual reproduction. They also move, and their movement has simple flagellum, if that is present. And then again, the most important thing, other than how do they live? How do they take their energy from? So we have mitochondria. They do not have a mitochondria. So bacteria do not have a mitochondria, but how would they supply themselves with oxygen? So they do it via cytoplasmic membrane. So this is kind of major thing. And also keep in mind that this slide will come again when I talk of fungi, because we have antifungal drugs. When we have some of the side effects that we have, if you were to use one particular antibiotic that's going to target your ribosome, or it's going to target your particular area of the cell, you may have a dual action. We normally divide bacteria based upon the external feature. How do they look like on petri dishes? And how do they grow? And what are the metabolic requirements for us to supply them with? And then we also look at the antigens, because we want to run a serology. And these days, as you saw yesterday, I was talking about microbiome. We can basically look at their genotype. So there are some advantages and disadvantages in each one of them. So let's talk about how do we look them externally with the naked eye. It's called macroscopic distinction. So we grow them on a plate, and basically they grow in colonies, and you'll see in the lab as well. They have a tendency to grow in colonies, and one colony may have millions and billions of cells, and we then look at the color of the colony, size of the colony, shape of the colony, and smell of the colony, because once they divide, remember I told you the odor that comes from the skin and the odor that comes from the mouth is because of the metabolic activity of the bacteria that's taking place. Now, why do we have to look at the external features of this bacteria? Because when we do antibiotic susceptibility test, we basically see if our antibiotics have removed those bacteria or killed them. And some bacteria, if there are more than one bacteria, we want to distinguish one form from the other. So we change their metabolic requirements. So for example, we may or may not provide whatever they need. For example, like sugars. Some of the bacteria are sugar fermenters. We call them lactose fermenters. And some of them will be non-lactose fermenters. And some other bacteria have a very specific requirement. And remember, as I said yesterday, those requirements can be stopped. If you want to go after them, you want to stop their multiplication, you want to stop their growth. That's the basis of antibiotics. So most of the antibiotics that we're using today are going to go after the bacterial growth. So we need to know how do they grow. Once you have an infection, bacteria goes into the system, they release toxins, they release many different enzymes. So sometimes they cause tissue injury, sometimes they cause anemia. So these are some properties that these bacteria have, and you can see they may have hemolytic properties. They lyse our erythrocytes. So when you get infection, you lose your red blood cells. And if you lose your red blood cells, your ability to provide oxygen to your body will be compromised. Sometimes they have lipases, a set of enzymes like Clostridia. It has a lipase that is going to cause tissue necrosis. So the major problem in these cases is tissue injury. And again, if you want to grow them in the petri dishes, you have to provide them whatever they need. We normally use a very rough method of dividing bacteria we call gram stain, and you will do that on Monday, those of you who are coming, called gram positive and gram negative. So we have antibiotics for the gram positive bacteria, antibiotics for the gram negative. So if you were to look under a microscope, how do they look like? Because they are less than 5 micrometer. You may be able to see a colony, but if you look at the microscope, which you will look at when you come for the lab, we look at their shape, we look at whether they are circular, longitudinal, what's the shape like? And then we also look how are they arranged, because sometimes they tend to arrange in pairs. We call diplococci. Sometimes they tend to arrange in a fashion which may sound to you like a bunch of grapes or a chain. So the names are coming from their appearance. Most of them, let's begin with the circular ones. They will appear circular. So they are like tiny little dots. And we call them cocci, or one single coccus. So you have Staphylococcus. So that basically means it is a coccus. It's a circular bacteria. They can also come in pairs, like Diplococcus. The typical example is Strep pneumoniae and Neisseria. So if you look under the microscope, they will tend to occur like pairs, as compared to Streptococcus, which will appear like a chain. Strepto basically means a chain, and coco basically is the circular part. So C for coco. They may appear like bunch of grapes, like irregular grapes. That's where the staph word is coming from, the Staphylococci or Staphylococcus. And methicillin-resistant Staph aureus is one of the important bacteria that we are dealing clinically these days. They may appear like a rod. So rods are bacilli. They appear like a rod, and they are bacilli. The typical example for is Escherichia coli or Streptobacillus. And sometimes we may see half of them in a cocci form, half of them in bacilli form, and they kind of irregular. The phenotype is irregular. So we call them coccobacilli. So you have like a mixture between cocci and bacilli. And some of them are spiral, like a typical spiral spaghetti kind of a thing. It's like a typical example is Vibrio cholera, or if they are very thick like spirillum, we call it treponeme, or they can be spirochetes. So these are typical examples of some of the bacteria that appear like coiled, right? From one coil to more than one coil. And sometimes they even appear like a worm, but they are bacteria under the microscope. And these are the classical examples. So I would want you to remember classical example in each and every group, because these are important bacteria that we'll take them anyway when we go and discuss that. Now, as I said earlier, we train everybody, every medical student, pharmacy student, dental student, they all are trained for gram staining. Why? Because it is a very rapid, powerful test to differentiate between gram positive and gram negative. Because we want to initiate empirically therapy. You don't want to give both to the patients, remember, for gram positive as gram negative. So we are pretty much safe if you give them one particular therapy to initiate therapy and then wait for the results. So that's one big advantage of gram staining, and you will do it yourself. It's pretty simple. It's based upon a dye, and one of the dye gets trapped, the other dye doesn't get trapped, so we have to recolorize it. So that's the basic principle of gram staining. We'll discuss that in detail, but for now, if you were to look under the microscope, gram positive will appear purple. So P for purple, P for positive. And those bacteria which do not retain that purple dye, they basically have to be counter-stained. So we counter-stain them with a dye called safranin, and that appears red. So gram negative or red. Basic scheme you can see, you do that in detail. Gram positive, gram negative. Classical example, Staph aureus, Escherichia coli. And then you can stain them. It is going to be purple as compared to red. And these are the different forms, as I just mentioned. Coccus, Bacillus, Coccobacillus, fusiform, Vibrio, Spirillum, and spirochetes. So you can see bacteria come in different forms. Now, I think most of the time I'm going to spend today is that I want you to know for sure what is the difference between a gram-positive bacteria and a gram-negative bacteria, right? And based upon your knowledge of immunology, which of these you think is going to initiate or provoke a disastrous response or a very harmful or pathogenic response? Positive or negative? How many of you say positive? None of you. So you only one is negative. Because gram negative is a big problem. Doesn't mean that positive is not, but gram negative is a big problem, and we're going to discuss that in a while. So you can see from here that we need to know their structure of cell wall. Why? The reason is because that's the very first barrier we have to cross. That's the barrier the antibiotics have to act on. So roughly, if you haven't begun studying antibiotics basically are working on cell wall. So we want to stop cell wall synthesis. That's the pretty much idea is. But we need to know what's happening. So you can see from here, gram positive and gram positive, again, typical cell bilipid membrane. So you have a bilipid membrane over here and a bilipid membrane over here. I usually give an example. It's just like you go and buy yourself let me give you an example for that. It's just like a slice of bread. So you can take a slice of bread and put like a cream on top of that. Or you can have a sandwich. You can have one loaf and then something in the middle and one thing other on the top. So like whole big sandwich. So the real sandwich in which there's a cream in between, that's a classical example of gram negative as compared to the gram positive, which is just one basic layer and there's a bunch of layer on top of that. We call that peptidoglycan layer. And then, of course, these are not the only things. There are many things which are protruding out of that. But if you compare gram-positive and gram-negative, and you can also make I would want you to make a chart as well. There are many charts available, but it should be actually in your mind that this one over here on the top is a peptidoglycan layer, a big topping, peptidoglycan layer. It is also present over here, but it is not that thick. So that's like a major difference that we have in terms of these two. And then again, we have other important structures. I just want you to remember two for now: teichoic acid and lipoteichoic acids. These are something which are jutting out of that, and a bunch of proteins. But if you look at over here, the most important thing, other than the cytoplasmic membrane, which pretty much looks like simple and same, there is another layer. As I give an example for the complete sandwich or toast, there is a outer membrane. So outer membrane pretty much looks like the cytoplasmic membrane, number one. Number two is there's a potential space between these two layers, which is called periplasmic space. And that's where many different structures are there, and we are going to talk about that. Because remember, if you're going to give an antibiotics will just have to pass through this layer, as compared to antibiotics in this case, which have to pass in both. So that will be a challenge for you to know. Then again, lipopolysaccharides, these are some of the molecules which basically are jutting out of the outer membrane, and there are a bunch of antigens over here, bunch of proteins over here. Some of the proteins are protruding through and through, and some of the proteins are just lying in these periplasmic space. Okay? Now, if you look at the structure of peptidoglycan, that's the most important structure. You can see from here A, B, and C. And this basically is typical structure from Staph aureus, Staphylococcus aureus. So A basically is a mesh-like structure, which is the outermost structure we call peptidoglycan. And then again, B has another mesh, and you can see over here, and it's a repetitive mesh where you have a polysaccharide polymer, and this is cross-linked. You can see these links over here by peptide bonds. And if you look at number C over here all the way through, these are again peptides which are cross-linked through a peptide bond, and these are important if you pay attention over here, because you need to know that before you understand how would a antibiotic for gram-positive bacteria act, because those antibiotics are going to break these bonds. So you have a peptide bond between the terminal D alanine, right, from one chain, and a lysine from the other. You can see lysine from the air, and this is all the way bound to alanine over here. So these are some of the typical chain, and in this case we call it pentaglycine bridge, which is Gly5, and that basically expands through and through when they want to cross-link in Staphylococcus aureus. So any drug which is going to act on the cell wall is going to break, because remember, there's one thing you have to keep in mind when we teach you antibiotics. Some of the antibiotics are, we call bactericidal, so they are going to break bacteria, and some of them are bacteriostatic. They will stop the growth of bacteria. So bacteria are continuously dividing, so we want to go and have our drugs over there that will basically break these bonds. That's why we have a choice. Some of the drugs will act over here, some of the drugs will act over here, some of the drugs will break the bond, because we don't want bacteria to synthesize and maintain cell wall. Okay? Now, also keep in mind that gram-positive, gram-negative is not the only criteria for bacterial classification. We have some bacteria that may not stain with gram-positive and gram-negative. So we have to go for some other method to stain them. Typical example is mycobacteria. And you can see these are like bacterial exceptions. What is happening in mycobacteria is that it has a special attachment of some of the factors which are covalently attached to arabinogalactan. Because once we teach you the drugs for TB therapy, because TB drug mycobacterial tuberculosis is different than gram-positive, so it has very specific molecules or specific factors which are there for us to target, right? In this case, arabinogalactan, cord factor, VAX-D, and sulfolipids are important. And they will also stop them from staining with gram-positive because it's complicated. So when we say mycobacterium, the more complicated the cell wall is, the more complicated will be the treatment. You agree with that? That's what happens. That's why anti-TB therapy is tough. Okay? If I was to ask you gram-positive, gram-negative, and mycobacteria, which one will have a difficult set of antibiotics and difficult set of treatment regimen? Mycobacteria, correct? They usually give like 6 months therapy, 9 months therapy. It's a multi-drug therapy. So you can imagine still we cannot take control of that because they're complex. So we call these bacteria acid-fast staining. And their outer wall basically is responsible for the damage they do. And they also have anti-phagocytic capability. So how would that help bacteria? Because if they have a sticky lipid-like outermost wall, so they would not allow the phagocytes to come and phagocytose them and kill them. Now, these bacteria over here produce a specific acid we call mycolic acid, right? And there are two more bacteria, Corynebacterium nocardia, they also produce mycolic acid. One exception, very common asked question is that mycoplasma, and make sure that you know difference between mycoplasma, mycobacteria. They are two different bacteria. So mycoplasma lacks peptidoglycan cell wall, and it incorporates steroid from the host into their membranes. Because sometimes if they have a difficult or complex outer structures, we will have a challenge to penetrate that and use our drugs. So I gave you an example for acid-fast stain. Cannot be classified by gram staining. Mycobacteria and mycoplasma are the example. If you were to put all these together, gram-positive, gram-negative, and I would encourage you to do that because if you don't understand the cell wall structure, you will have a terrible time to understand the drug therapy. Because the drug therapy for gram-positive versus gram-negative versus mycobacterium is totally different. Now, this is a good picture over here. You can see gram-positive. All of them should have a cell membrane. So you can see the cell membrane pretty much is there. We have a thick peptidoglycan layer, and then we have lipoproteins in there, and we have lipoteichoic acid. I told you two important acids, teichoic acid and lipoteichoic acids. They basically are hallmark of gram-positive bacteria. If you look at gram-negative bacteria, again, there are two membranes. One membrane, same as this one, and another membrane we call outer membrane. So outer membrane is also only present in gram-negative. And then we have a thin layer for peptidoglycan. It does have, but it's a thin layer as compared to gram-positive. It would also suggest to you that the antibiotics that we use for gram-positive may not be used for gram-negative. That's what it tells you. And of course, none of these are going to work for mycobacterium because it's very complicated. Then we have LPS over here. You just answered me that gram-negative will cause a fulminant or dangerous immune response when it comes to dealing with bacteria. So the question comes, what is it in gram-negative bacteria that you think will cause that fulminant immune response? What is it as a part of gram-negative bacteria which is different than gram-positive bacteria that will create a havoc in our immune system? Sorry? Endotoxin, correct. And most of the time the answer is there on the slide, you just have to pay attention. LPS, lipopolysaccharide, is an endotoxin. Exactly correct, endotoxin, lipopolysaccharide. Because remember when I said endotoxin, I said the most powerful stimulant of immune response. It's going to cause endotoxemia. So that is the major problem over here, and you can see that. Okay. The other important thing is that in gram-negative we have pores because of porine proteins. When it comes to mycobacteria, we have mycolic acid, I just told you, glycolipids, lipoarabinomannan, and we have a very small peptidoglycan layer, and then many other molecules. So this is a very rough picture that I have for comparison. I would want you to make a little bit detailed one. Any questions on that? Any questions on this one? Okay. Now, one of the qualities that some of the bacteria have, that they form spores. You remember when there was issue with the anthrax, and you heard that they were putting powder in the envelopes and being posted to the people. So some of the bacteria can form spores. So these spores are important because they are resistant to many things that we normally have in the environment. So your antibiotics would not work. So spore forming bacteria are again dangerous classification. They are never gram-negative. So gram-negatives cannot form spore. That's a good thing. So if I say anthrax, I'm talking about Bacillus anthracis. That's what we have for the anthrax that form the spore. I usually give this example. Can we help with somebody's head? Yeah, okay. So you see somebody many a time wearing scrubs. And I see many people over here walking around in the classroom, going around in scrubs. Okay, the rule in the medical school is scrubs are only for in-room use. So, for example, the scrubs that you wear for surgery are to be taken out, worn in the surgery room, disposed of over there. You cannot be walking with the scrubs, right? Because scrubs basically are not washed. If you don't know that, I know maybe you guys are a different kind of scrubs. Scrubs are autoclaved. Scrubs are autoclaved. So the reason is that if your scrubs bear spores, for example, you pick up spores from the seeds and walk out in operation, they will not firstly allow you to do that, right? There are strict rules and regulations. So spore basically are something that are very dangerous. You cannot wash them with any kind of antibiotics. So you have to autoclave them. Autoclave is heat to that certain temperature with pressure to basically disrupt the bacteria. So you can see from here, Bacillus anthracis, that was a big challenge for the anthrax because it forms pores. It stays forever. So if you come across with the spores, you're in big trouble. And Clostridium as well. So Clostridium, that's what you call tetanus toxoid, roadside accident, because it's very common and it will stay over there. It is a part of soil bacteria. Now, how does, well, again, for bacteria, it is a homeostatic mechanism because if the environment gets nasty and the bacteria thinks that it's time for me to go in hiding or hibernate, so to speak. So what they do is that they actually take on this special cover. We call it a spore. So you can see it has a central core, it has a inner membrane, this is a spore wall, cortex, outer membrane, protein core, and exporium. So now you have a challenge. It's a big armor on the bacteria. Bacteria has shrunk, it has reduced its metabolic requirements, and now it is ready to go anywhere it feels like and stay forever in the system. So spore forming bacteria, very dangerous, and that's what you want to make sure when you open up people in operation theater. You don't want to put your scrubs, your gloves, your spores on the patient, and the patient has that problem. So you really have to have a very high level of sterility, very high level of caution when you deal with the spores. I don't know if any of you worked as a surgical assistant, because I remember when we were trained for surgery, we would not be allowed less than 15 minutes. So we have to wash our hand with scrub, with soap and water for at least 15 minutes before they would allow us to put on our gloves, exactly by clock. So you can imagine a serious issue when you go for putting on, patient for surgery. Okay, and most of the problem is coming from spores. Now, as I said, this is a phenomena where if bacteria wants to go and hibernate, vegetative state or dormant state, we call the nutritional requirements will be less, and basically it's a multi-shelled, that's what you saw in the case, and dehydrated. But the good thing for bacteria is that bacteria make sure that it has complete copy of chromosome. And why would it need that? To divide. Okay? And this is the bare minimum essential for this. And the most important thing that we need to know, because we need to come up with drugs that will even go after spores. So they have a high concentration of calcium bound to lipocolonic acid. So that's something that we need to know, because we do have some drugs that will go after them. But that will be a challenge for us. Okay. Now, if you saw the outer core, and if you were to look under the light under the microscope, that's kind of a shiny thing. So you will see that a shining thing because it is a protein keratin-like coat. And then the whole idea is that genomic DNA is protected from intense heat, radiation, and enzymes. So you can imagine how much temperature you have to achieve, at what pressure to get rid of spores. That's why we have autoclaves. That's why we want to autoclave surgical instruments. We need to go for that. So that's a higher form of getting rid of spores. Okay. Now, as I said earlier, decontamination will be a major issue if you deal with the spores. These spores can exist for centuries. And what we need to do now is that we have to come up, there are many ways to kill bacteria. For example, if bacteria needs a certain kind of food, because they are kind of, have very important nutritional requirements for them to divide. So one of the things that we do, or the drugs do, it will stop those nutritional requirements so they can stop dividing. But we need to know what are their nutritional requirements. Some would like carbohydrates, some would like blood, so on and so forth. So whatever they like, we provide them. Like for example, some of the bacteria have hemolytic properties. So this means they need to chew on the protein of red blood cell. So if you have to grow them in the lab, you have to give them blood in the petri dishes so that they can start dividing. They'll have these kind of fastidious nutritional requirements. And in this case, since we know that a particular acid like lipocolonic acid or calcium is required, so we have to come up with some of the drugs that will stop the supply to the spores so we can get rid of that. Okay? Now, we can also divide bacteria in terms of metabolic distinction. Remember, as a rule, I said that those bacteria that become your commensal in the airways, they are aerobes. They want to divide in oxygen. And those which can survive without oxygen, we call anaerobes. And then there are quite a few in between. So aerobes are one they like air, anaerobes are the one they don't need. Because the whole idea is that they have this capability for a specific nutrient to ferment specific carbs or use different compounds as a source of carbs. So you have to have a source of carb for generating oxygen in Krebs cycle. So you have this ETC there, electron transmission chain. So you have to provide them with the carbon source. So they can either take that from different places. They also produce characteristic metabolic products, and that's why bacteria are used in food industry as well. For alcohol, for example, for fermentation, your cheese, your curd, many different things, bacteria are used in fermentation, and they produce those metabolics that are required. So acids and alcohols and many different things are taken from these bacteria. Now, we can also differentiate bacteria based upon the antigens that they carry. And of course, if they have an antigen, we have an antibody. So that's a rule. Okay? If you have an antigen, we have an antibody. That's pretty much the standard for diagnosis of bacterial infections. And again, as I said, these are quick, rapid diagnostic tests. We call it serology, and these tests are called serological tests. So, but having said that, the reason is that why do we need to go for serology? Because some of the bacteria would not divide in nutrient agar, or would not meet the criteria that they need to divide over a period of time, because they may take a long time. Like, for example, some of the bacteria, we call it doubling time, they can double within seconds. So they start with 1 million, within a second they are 2 million. So there is a rapid multiplicative progression of bacteria. But some of the bacteria will take months because we are unable, or they are unable to gain what is required. So these are some of the lists of the bacteria. For example, Treponema pallidum, causative agent of what? Syphilis. Sorry? Yeah, syphilis. We don't want to give time to Googlers, because if I give more time, I can see the Googlers will jump in and just take unnecessary credit. Okay, it's on the slide. There you go. Okay. Then some of them are too dangerous for us to grow in the lab. Francisella, because you don't want to grow something in the lab and then gets out of hand. Okay? And some of them have specific diseases like Escherichia coli, that's responsible for hemorrhagic colitis, and you see every month there's a report on the news, breakdown of one of the Escherichia coli strain. And then again, we want to do that ASAP because of the sore throat. Remember I said that the strep throat is dangerous because of problems, especially when it occurs in the babies. So these are some of the things that are required. Now, if you to go for the genes of this bacteria, the techniques are DNA hybridization, polymerase chain reaction, and amplification. The good thing for these genetic methods are that they will pick up living or dead bacteria. So that's good and bad as well. For example, bad in the sense that you pick up a bacteria and the bacteria is dead. What's the point? Right? Good in a sense, you just need 1 or 2 organisms and you can pick them up and stain them. Right? So what we do is that we can use them as a rapid detection method. We can also use them for some of the bacteria which are slow growing and they take months to grow. For example, Mycobacterium will divide in a month. So one Mycobacterium will divide no less than a month into 2. So that's another reason. And we can also use these genetic distinction techniques for epidemiological investigations. For example, if meat is supplied by one company in Florida to 50 different states and there is an outbreak, so we will go and genotype that particular bacteria and link it to the source. So we can do that only with the genotype. So that's what advantage is, that you can basically use that. And as I said, what we normally do is we use plasmid analysis. I'm going to talk about plasmid, very important. Ribotyping. What do you understand by ribotyping? Ribotyping. Ribotyping. Correct. Of course, simple, ribosome DNA. Ribosome, you want to type ribosomal DNA because that is the major stuff over there that our antibiotics are going to target. Most of our antibiotics are going to target that specific ribosome. Correct. Okay. Now, DNA, again, is the secret here. The cytoplasm of the bacterial cell wall contains DNA chromosome, messenger RNA, ribosome, protein, and metabolites. So that's it. If you were to ask what is a bacterial chromosome, so the answer is it is a single and it is double-stranded circle. So it's a double-stranded circle like a chain. It is not contained in the nucleus because they don't have a nucleus. And it's usually detectable in an area which looks like nucleus. We call nucleoid. Nucleoid looks like nucleus because there's a condensation of genetic material there. And if you know about DNA, histones are there, and I have a slide coming up, I'll show that to you. Histones are not present in bacteria. Okay? And then finally, DNA does not form nucleosomes, so nucleosomes are not formed. See some of the distinguishing characteristic of bacterial genetic material. And this is important for you, many times asked, because we will discuss in the next lecture about the antibiotic resistance and how that happens. What are plasmids? So plasmids are small, they are circular, and they call extra chromosomal DNAs, very unique for bacteria. And again, they are found in problematic bacteria like gram-negative bacteria. The question that bothers most of the microbiologists is that are they required for bacteria to have it? Why would bacteria have plasmids? But it's good for us. I'll explain that to you in a while, because that is where the bacteria have a selective advantage. So they're like 100 bacteria, and you use antibiotic, 90 will be gone, 10 will survive. That's the survival of the fittest. Darwinian kind of way of doing it. The reason is that there's a bit of information that these bacteria carry and contain. Not only they carry, contain, but they would pass it on to other people as well. So this bit of information comes from plasmids. One of the important plasmid that I'm going to discuss today is for fertility, because this is going to pass this information. Remember, I just said that bacteria divide by binary fusion. So one divides, so they call it daughter separation. So one bacteria, two daughters. One bacteria, so it's going to be a typical multiplicative progress we call binary fission. But at the same time, we have this specific plasmid that has selective advantage. So if a bacteria has that plasmid, we call it F positive, fertility positive. And this is the major thing that you need to know if you need to understand why is there antibiotic resistance, why your antibiotic is not acting, why a toxin is produced by that, and so on and so forth. So the plasmid, especially F, stands for fertility. I'll come back to it later. So we already discussed that there is no nuclear membrane, but the question you have to ask yourself, well, why would bacteria not have a nuclear membrane? The reason is because bacteria, since it has survived for trillions of years, has for some reason maintained a very simple requirement for synthesis of protein. It has to make proteins for sure. And actually bacteria are making proteins for us as well. Your insulin, your many drugs, who makes it? Bacteria. So you put the sequence of that into bacteria, and bacteria makes insulin for you and makes hormones for you. So bacteria, we use bacteria for a good cause. Okay? So, and then again, it's very simple. You just have to insert it into that genome, and it's going to multiply, and we can then separate it. So transcription and translation are coupled. That's another important thing in bacteria. Ribosomes can bind to messenger RNA, and proteins can be made as messenger RNA is being synthesized. So it's going to still attach to a DNA, need not to be separated, and it can initiate the sequence of protein synthesis. So all these proteins, like you talk of hormones, you talk of cytokines, so on and so forth, good and bad, can be made by bacteria. Okay, so that's very important concept you have to have simple requirement, but it's a dangerous for as well because we will have a challenge to go after them and kill them. Ribosome, and very favorite question, there's a difference between human ribosome, a plant ribosome, fungal ribosome as compared to, and we need to know the important aspect of even subunits. It contains a 30S plus 50S. It forms a 70S ribosome. Whereas we humans have an 80S, this means 40S per 60S. So thing is this, if you are going to use a drug which is going to target a ribosome, you want to make sure it targets bacterial ribosome, not your ribosome. So it's good that they are different, okay? Now in this case, all ribosomes are free, none are bound. So that's the other important, and as I said earlier, these ribosomes are the major targets of antibacterial drugs that we have in the market. If you look at the further details of cytoplasmic membrane, I would encourage you, I'm going to show you a table. Murray's textbook has a beautiful table, and this is like a cramming material for you. And it has transferred proteins, it has ionic pumps, because what may happen is if you give an antibiotic to kill gram-negative bacteria, and it has a pump there, it would not allow the antibiotics to pass through, it's going to pump it out. So that's what we have problem in terms of use of these antibiotics. And then again, these structure of bacteria like cocci and bacilli are kept by protein we call actin-like proteins. So there are, because remember when we go to kill bacteria, do you need to disrupt their cell membrane, cell wall, nucleus, ribosome, everything. And this is what I was talking about. And you can make your own, pretty much the information that we already discussed. A cell wall versus a plasma membrane gram-positive, gram-negative, and other structures. So you can see the important structure and the chemical constituents. So that's the cramming material for you, because when we teach you the drugs, so drugs will target, for example, one of the drug will just target, let's say, teichoic acid over here. And is going to go after this specific cross-linking to peptidoglycan. And that's like a mechanism of action of that drug. Right. So you have like at least 10 different drugs for, and each one of them will target. And that also for those of you who are interested in research and development as well, because you have to come up with another structure to go after that kill. So that's a good thing for you to remember, earlier the better. Again, function of bacterial envelope, you see the outermost things which are there. For bacteria, it gives protection, it gives like a antigenic response, and then again, this is something that you want to penetrate into before you go for that. So you can see that it gives rigidity, it has a packing of internal, and then these are the bacterial function, is a permeability barrier, anything that needs to go up and down has to pass through that. Energy production is by membrane, then it has a motility flagella. For flagella, this is like some of the bacteria which will have a tail, a navy tail, like swimming. So you just kind of move the flippers. So you will move forward. So bacteria, when it goes to the blood, it's going to swim around. It has this flagella, push forward and cause more problems. So bacteria which have pili, one of the pili bacteria has is for conjugation. I'm going to discuss that a while. I did say there's an asexual, but there is a sexual production, reproduction in bacteria as well. We call conjugation, where they actually mate, and there is a male bacteria and a female bacteria, and the genetic material is passed from male to female, and that pili is called mating pili. And then remember I said that the bacteria had to adhere to epithelium, it has to get attached, it has to go and respond to immune system, and so on and so forth. So each and everything that is there for bacteria, we need to know. And many things which are out there, especially in the outer membrane, if it's especially a protein, so we will have lipids for that. Okay, this is a comparison. You can read it on your own. Let me go to the last slide. Okay, now what I want you to do is that those two tables, try to understand that and try to make sure, because the idea over here is that we need to have a very clear vision at this stage, especially for this lecture, what is the difference in the structure in the three kinds of bacteria. And each structure is affiliated with a function. And then again, we need to target them. And that will give us a choice, because the whole idea is if bacteria is making peptidoglycan, we need to know how it is synthesized so that we can break it, right? If a bacteria is making like lactic acids, these joining material, we want to disrupt the structure of the bacteria. So we want to go for each and every particular molecule that it has, okay? But at the same time, also keep in mind that anything that's jutting out of the outer structure as a flag is basically an epitope for immune response, and immune response is going to go. We call them basically, some of the proteins for bacteria, we call them like surface proteins. And we have a very specific response for that surface protein, okay? And there are one or two structures, again, I will talk about that later as well, but for this part, we just talked about lipopolysaccharide, endotoxin. The structure of endotoxin, very commonly asked question. And you can see over here, different components, LPS, A component, B component. I'm not gonna go in details to ask you to memorize the sequence over here, but I would definitely want you to know the difference in terms of different segments and what are their antigens. For example, O antigen versus a core polypeptide. The reason is, if you have an O antigen, we will have an O antigen antibody. There's going to be a diagnostic test for that. Okay, so it's important for lipopolysaccharide. So spend a little bit of time on the structure for lipopolysaccharide. We call it endotoxin. And again, keep in mind that this is an important that will enhance the immune response. One other thing that usually gets confused with lipopolysaccharide in terms of the structure of LPS is basically lipo-oligosaccharide. We call LOS. So LPS and LOS are two different things. So also keep in mind, some bacteria may have one, or the other bacteria will have an other one. Okay? So these are some of the important things generally that I alluded to for this lecture. But again, keep in mind that once we go in detail out, we'll talk about each and every separately. But for now, at least you can start working on the structure of cell wall, the capsule, and I would rather encourage you to keep yourself to some of the slides which are there, and that should be okay for you. They are from the book, but if you want to, for cramming material, that's good enough. But conceptual things, what I would suggest to you is that try to understand the importance of it. For example, why is it that LPS is going to cause immune reaction? Right? Why is it that endotoxin is bad? Why is it that we have a problem with treating gram-negative bacteria? Why is it that we have a challenge in taking care of mycobacteria? Things of that sort. I think it will help you form the basis for that. Okay? And thank you, and sorry for running a couple of minutes over my time.